Hi, this is Scott Ferguson at SUNY Fredonia. I'm here to teach you a little bit about ImageJ. This is some software from the National Institutes of Health used to analyze imagery from microscopy to gel electrophoresis. So today we're going to learn about analyzing a gel consisting of two different channels, molecular weight standards and unknowns, and learn how to interpolate the molecular weight of those unknowns based on that latter. So what we're going to do is start by opening up a file. Open, and we're going to take a look at a file that is a pilot that I did for the experiment that we're going to do in lab this semester, and you can see that there are red and green channels. First thing that we're going to need to do is split those channels into what's called a composite, because it's actually two different images on top of each other, the green image and the red image, which were acquired simultaneously. So once we click that composite button, you'll see that we can turn off and on the different channels. And so this is the red channel, this is the green channel. They contain different data, and they while they overlap in molecular weight, one does not affect the other. They're independent signals. So we're going to leave that as such. You'll know that you've done it correctly if you see this little designation at the top of the image. If you roll your mouse wheel, you can see that you can switch between channels. That's also this slider at the bottom of the image. Click on that, that goes to green, and there's no blue data, but it is possible to have 3 or more channels that would be designated red, green, and blue. So when we look at our gel here, you can see it's kind of long relative to how tall it is, and so if we want to see things a little bit closer, you can press Control+, and that will zoom in a little bit so you can see a bit more detail in your gel. To navigate around within the gel, you'll see this little purple box up here. There's a box within a box, and what that is the smaller box is what you're looking at that moment, and the larger box is the entire image. To move around within there, come over to your original ImageJ toolbar, select the hand, and you can see that you can drag that image around. We're going to start at the top of the gel, and what we have to do is select the different lanes that are in our gel. So we'll designate one lane from the other. As far as ImageJ knows, at this point it's just a picture. It doesn't know where the lanes are, and it doesn't know where the bands are, so we have to specify that information. So what you can see is I'm drawing a square. I've selected this square tool over here, and we want the selection to be narrower than the band itself, narrower than the lane, that is. And the idea here is that you don't want to have any signal contaminating from the lane next door, so you're going to pick the center of each lane. I've gone over and selected the hand tool again so I can get to the bottom of the image, back to the rectangle, and then you can grab one of these little handles and stretch it down closer to the bottom. It's unnecessary to incorporate all this stuff at the bottom. What this is unincorporated dye and primers that were not participating in the reaction. So these are not anything of interest to us to analyze. So now that we've selected this entire lane, what we have to do is designate it as a lane in the software. Go over to the Analyze menu here, down to Gels, select first lane. You see this little 1 appears. Now we're going to drag this box over to one of the other lanes. It's very important that you don't click outside that box. If you see my cursor is turned to a plus, whereas when I'm in the box it's an arrow, you want it as an arrow. So we're going to drag this box over here. The position up and down doesn't matter, because ImageJ will automatically snap it back to the same up and down position. So we're going to center it on this lane here, go back to Analyze, Gels, and select Next Lane. You can see it snapped down so it's perfectly aligned. Move over here, Gels, select Next Lane, and there's a shortcut for this, which is Control+2, and so I'm going to just do that and select the remaining lanes in this group of samples here. Okay, so we've got 11 lanes here. Now, this is all well and good, but in order to determine the molecular weight of these samples, what we have to do is generate a profile of the lanes. And you want to make sure that you're selecting the red channel at this point. If you're not, you can slide this slider back and forth until it gets to red. All the way to the left is red. I'm going to go to Analyze, Gels, Plot Lanes. Now you get this gigantic window here, which consists of a space here. This is a lane where along the x-axis you have the position on the gel. Left is the top, right is the bottom. And then on the y-axis, up and down, is the intensity of bands. We scroll down here to a lane where there's actually bands. You can see these peaks. These peaks each correspond to a band. A tall band like this is more intense than a short band like this. Now this is only the data from the red channel. We have to go back to our gel and plot the green data. So you can see that I'm still selecting red. I'm going to slide this over until it's green at the top here. Analyze, Gels, and you want to press Replot Lanes. If you press Plot Lanes a second time, you'll get an error. So it's Replot Lanes. And you can see if we compare these two plots, they're different. This first lane has peaks in the green plot, whereas this one doesn't. That's because our molecular weight standards are in the green channel. We're going to do some conversions of this so we can overlay these two plots and see them simultaneously. For each of these images, we're going to go to Image, Edit, and Invert. So that just changes the background to black and the signal to white. I'll do that for the same thing. So Edit and Invert. So now we've got these two samples. Now we have to combine them. Go to Image, Stacks, and then this selection here called Images to Stack. What that does is it's going to combine those two images together into a single file. You don't need to keep the original sources, although you can. Go back here, and now we've got this tall thing. I'm going to zoom back in on it, where if we go to the top, you can see that this is 1 of 2. If you roll your mouse button, it changes. And so that change refers to this is the green channel and this is the red channel. Right now they look black and white, so we need to do some further manipulation to get them into the red and green. So you're going to go to Image, Color, Stack to RGB. And that's going to take this data and replot it overlaid like this so that we have an overlay of the red and green data. This is the format in which you want to keep your image so that you can analyze molecular weights. You can close that other plot. Now is a good time to save. So save as. TIFF file will keep all of the information that we need. So this is going to be demo stack. So we're going to zoom in a little bit here. Make our window a little wider so we can see all the data. So this first lane here is our molecular weight standards. If we go back to our original picture, lane 1, this is lane 1 on top. Remember, left is the top of the gel, right is the bottom of the gel, so going down this way. And then our lanes proceed left to right in the gel and top to bottom in the plot. So the first thing we need to do is determine the positions of these molecular weight standards. Each of these peaks corresponds to one of the standards, and we're going to collect the position by clicking on this little point selection tool here.